This conference will now be recorded.
Okay, folks, so good evening, and I'm glad you're here, and we are going to be going over the second study guide and important information regarding the skeletal muscle contraction, and so the muscular system. And so last class we discussed and we went over the actual components of skeletal muscle, and we talked about and we looked at the connective tissue coverings as well as the bundles of structures. Okay, we'll do that before I move on, but I want to just, since I was mentioning this before we started recording, I just want you to see how, as we're looking at the neuromuscular junction, I said to you that this is presynaptic, the axon terminal, postsynaptic at the motor end plate of the muscle, and in this space right here, this is called a chemical synapse. So we have a synaptic cleft, this space where the chemicals, the ligand, the acetylcholine, that neurotransmitter, will leave from the axon terminal and then have specific receptors on these postsynaptic ligand-gated channels. Okay, and a ligand, those are chemicals, right? So we have ligand-gated channels, we have voltage-gated channels over here. See that? And recall that when an action potential travels down the motor neuron, right, the axon of the motor neuron, and then it reaches the axon terminal, it will trigger these voltage-gated channels. These channels respond to voltage, they respond to electricity, the action potential. It acts as the key. That key will open up this channel and allow for calcium to enter in. When calcium enters into the axon terminal, it will cause these synaptic vesicles, which contain acetylcholine, to then release their neurotransmitter, acetylcholine, into the synaptic cleft. So electrical event, chemical synapse, then electrical event again. Okay, so very important to remember that and to see that. Presynaptic, postsynaptic. This is a good one also to share with you. I have a couple of images to share with you from on Friday's class, and I'll share this image also with you in documents and resources. Okay, I like this image, it's a pretty good image. All right, but really important to review your different types of channels, and that would be seen in chapter 3, I believe. Chapter 3, looking at the cellular structure. We talked about the plasma membrane, as well as we talked about channels and such. So please review that. So voltage-gated channels, ligand-gated channels, very important to remember and to know. And again, chemical is the key for the ligand-gated. Electricity and action potential is the key for voltage-gated. Okay. Let's see. Let's also then go to, I'm going to open up the study guide for one first, and just want to review as far as structures are concerned. And this image right here is a very good image, and just showing you again that these cylindrical structures comprise skeletal muscle. Recall that we have the different connective tissue coverings, epimysium, paramysium, and the endomysium. They will cover, the epimysium covers the muscle itself, the whole muscle structure. The paramysium covers the fascicles, and the actual endomysium is covering these muscle cells, aka muscle fibers, also known as muscle fibers. And then we have the cylindrical structure. So we have the fascicle. Fascicles are comprised of here's a fascicle. It's comprised of the muscle fibers. The muscle fibers are comprised of the myofiber. You see here, this is a muscle fiber. This whole structure right here, they're comprised of these myofibrils, right? Each of these cylindrical structures is a myofibril, and the myofibrils are composed of the myofilaments, actin, as you see here. Here's a myofibril, so actin and myosin. Actin thin, myosin thick myofilaments, okay? We also talked about the sarcomere going from Z disc to Z disc. And we talked about, and you see how the A band, right here, and here, and here, that's where there's just actin fibers. The A band has a combination of actin and myosin fibers, so there's a crossing over, so it creates more of a dark line, and this is what gives the striations for skeletal muscles. So remember, striations are present in skeletal muscle as well as cardiac muscle. Okay. So, and then just remembering the epimysium covers the muscle, perimysium covers the fascicle, and endomysium covers the muscle fibers. And then you'll see here sarcolemma. Well, what's that? Well, that's the plasma membrane of the muscle fiber, aka the muscle cell. Okay. Just review this and go over this, folks, please. And then just again, looking at really the very end result of what's going on as far as muscle contraction is concerned. We talked about isotonic and we talked about isometric contraction. So primarily you're thinking if I'm contracting a muscle, skeletal muscle, you're thinking of isotonic. The muscles will contract and they'll shrink. They'll as we contract, the muscle belly gets smaller, closer together, right? There's a contraction. That's the isotonic contraction. Isometric is when we have just the example right here of a cross bridge, when there's just a cross bridge. So if you were to just tighten up the muscle, but not shorten the muscle, not allow for the origin to come closer to the insertion, right? We talked about those terms. That would be isotonic. This is isometric. If you contract your abdominal muscles, your rectus abdominis, right? Contract that, what's going on? And you're not like doing a crunch, but you're just contracting it. That would be a isometric contraction, okay? We talked about calcium binding with troponin. Troponin will change shape, and because it's attached to tropomyosin, this line here, tropomyosin will move out of the way, exposing the myosin binding sites on actin. That's important that you remember that. Tropomyosin moves out of the way and exposes the myosin binding sites on actin. Okay. Now, let's see here. I wanted to review this with you also, just showing you that again, the sarcoplasmic reticulum, the SR, is this blue structure here. I kind of think of it as you would think of a like a sleeve, like a sweater, you know, that's kind of like a loose sweater, holes in it and such, or like a crocheted type of thing that's covering what's going on here as far as these structures are concerned. So you're seeing here, folks, that we have the muscle fiber with the myofibrils, and present within the myofibrils are the myofilaments, okay? And once the action potential travels along the sarcolemma, right, and travels down the T-tubule, then it will allow for then on the terminal cisternae of the sarcoplasmic reticulum, there are voltage-gated proteins present that will open, they'll change shape and open in response to the action potential. And as a result, they'll release what? Calcium. And once they release the calcium, that's going to interact with the troponin, allowing for myosin binding sites to be exposed, allowing for contraction, allowing for cross-bridge formation to occur. Okay, so, you know, you really should be reviewing those 3 videos and looking at all the steps regarding what takes place as far as skeletal muscle contraction is concerned. And by doing that and by writing down the notes and such, right, you're also remembering also the structures. So you're looking, you're focusing on the physiology, but you're learning the anatomy as you're focusing on the physiology of skeletal muscle contraction. Okay. And good. Okay. Yeah. And so I'm going to now begin with, yep. And so this is really important as far as the generation and propagation of the action potential. And again, when we get into looking at neurology, and once we stop looking at, so in neurology, we look at different aspects of the nervous system, as well as the different parts, components, and as well as the cells. And then we'll talk, we'll revisit action potential formation and such. But here we have, and this is very important to see, and this is why I started off the part 2 notes. You're seeing here as far as action potential of the sarcolemma. So again, remember, there we go. Remember that here we have presynaptic electrical event. At the synapse, in the synaptic cleft, we have a chemical event. And then on the postsynaptic, on the motor end plate of the skeletal muscle, we also have an electrical event that takes place, okay? And so you'll see here as far as the sarcolemma, the plasma membrane of the muscle fiber, the muscle cell, aka also known as. So you'll see here there's a local depolarization, begins at the motor end plate. Then a depolarization of the plasma membrane will occur of the sarcolemma, which will in turn allow for then this electrical event to spread away from the motor end plate and then spread along the sarcolemma, go deep into the T-tubules, and then interact with the terminal cisternae of the sarcoplasmic reticulum, allowing for release of calcium ions into the area where of the myofilaments, actin and myosin. And remember, actin slides upon myosin. Myosin is stationary other than the head pivoting, right? So the head of the, it looks kind of like a golf club, right? That pivots, but myosin is stationary. Actin slides upon myosin, right? And you saw that in the videos. It's very, very easy to see. So we recall that depolarization takes place first, then repolarization takes place. So depolarization, a change in the plasma membrane from the resting membrane potential. Remember that that's, what do we say here? Positive on the outside, negative on the inside, okay? As a result of higher concentration of sodium ions outside, higher concentration of potassium ions inside. So it's positive and negative, and that changes, that flips to negative on the outside, positive on the inside. That's a depolarization event. And then it goes back to, returns to the resting membrane potential via this repolarization, okay? So let's look here and review this. And so you'll see that we have the local depolarization. We said, and I said to you that, and I made an important note of saying this, that sodium ions always move first. That's really a key issue there, folks. Sodium ions are moving first. They're passing through, right? Depolarization takes place. Then we have, as far as repolarization, potassium ions moving. Now know that there are leak channels. Remember, recall that in video number 1, it talks about the leak channels. They're always open. There's no gate. There's no gate. They're always open. And so ions will travel down their concentration gradient from high concentration to low concentration. That's diffusion. High concentration to low concentration. It's a passive process. No energy is required. But to go from low concentration to high concentration, we think of, look down here, the sodium potassium pump. That's moving 3 sodium ions outward, 2 potassium ions inward. It's going against the concentration gradient. So it's an active transport. We need energy to do that because we're going against the concentration gradient. Okay. So please, review this and go over this and rewrite it out and such. Again, we're going to revisit this in neurology. So it's important that you grasp it and you watch many videos regarding it. Ask me any questions when we get back on campus and such. It's important. It's an important concept. It really is. Okay. And it's a little tricky, to be honest, right? And so you'll see here that, I mentioned to you, I mentioned to you the leak channels. I mentioned the ligand-gated channels, and I also mentioned the voltage-gated channels. So there are different types of channels, and they have specific receptors for specific ions and such, okay? But the key is remembering there's a local depolarization, then we have depolarization of the sarcolemma, that's the plasma membrane of the skeletal muscle, then we have a repolarization, a return to the resting membrane potential. Now, let's see here. Let me make this a little bit bigger. Okay, so now when I said to you, and I've said this before, and I'll repeat myself, that muscle contraction, when a muscle contracts, so think of the biceps brachii. That's just an easy one. You can just, we always thinking of that's an easy one to just look at and to realize that, hey, I see the muscle belly. The muscle is anchored via the tendons, right? And we know that when that muscle contracts, as far as it shortens, that's an isotonic contraction. When it doesn't shorten, but that there is a cross bridge formation, but no sliding of actin, then that's an isometric contraction, okay? And know that depending upon, and I said this to you before, that this is what I was thinking of, depending upon the load, okay, the load, meaning, so if I lift up my pen, that's pretty light, right? But my cell phone, is much heavier than my phone. And then I have even other stuff around here that are, you know, like even this glass is a little bit heavier, right? And so know that depending upon the load on the muscle, that's going to really determine, and your brain will help to figure this out, how many muscle fibers are engaged in contracting in order to move the load. So look at here. So contraction produces muscle tension, the force exerted on the load, right, whatever we're trying to hold, pull, or object to be moved. Okay, so, and know that again, like I said, not all the muscle fibers are contracting unless it's like a really, it's a heavy thing. So say I'm like 50 pounds and I need to like, you know, try and do a curl, which is pretty heavy, right? So like right now at this age, I can do like 35 max, you know, like and I used to do much more than that, but that's pretty heavy, right? And pretty much all my fibers in that biceps brachii and other muscles that are synergistic with that will be working to lift that load. You know, you could really feel the difference in comparison to, or how about if I were to put 100 pounds on there and try to do a curl, forget it. I would not be able to exert enough force and contraction in order to lift that as like a good curl, right? So that's an important concept to remember regarding skeletal muscle contraction. You'll see here each muscle is innervated by at least one motor nerve, right? And that's important also to see. And we saw, and if you look online and you see even with in the videos and such, you'll see that, you know, yes, the neuromuscular junction, it's going to have then really the interaction, that chemical synapse between all of the muscle fibers, aka the muscle cells, with branches from, right, the axon. So the axon is the extension of that motor nerve. Okay, and we would say, folks, that axon will carry information it's away from, right? It's leaving the motor nerve. Okay, it's not sending information back, so it's not sensory. Sensory would be sending information back to the brain. In particular, it's going to go to the thalamus, deep within the brain, and then go out to different areas of the brain for some type of reaction. We're talking about motor output, not sensory input. Now, next semester, we'll be talking about sensory input. Okay, we'll be spending some time regarding that, absolutely. But we're talking here about motor output, okay, motor neurons and such. And so the axon, so dendrites bring information to the neuron, axon takes information away from the neuron, okay? Now, so motor unit, the term motor unit consists of the motor neuron and all the fibers, the muscle fibers, from only a few to many that it actually supplies. So that's pretty cool, right? That's amazing that really, depending upon what's going on, this motor unit, it can be going to many fibers or only a few fibers, okay? You'll see here also that a twitch. So this is the simplest contraction. So when you think of a twitch, you've probably had like a twitch in your eye, right, of the musculature in facial muscle or the actual, yeah, more facial muscle, because you'll feel that like little twitch, and it could be as a result of some type of virus that you're experiencing and going through and just causing that. It's involuntary, but it's a simple contraction of just a very quick little twitch. Simplest contraction resulting from a muscle fiber's response to a single action potential from a motor neuron. Muscle contracts faster than it relaxes. That's important. It's smooth, and the strength varies with the need, the demand, the load that's placed upon it. Okay. Now, interesting here. So eye muscle contractions are rapid and brief. Okay, so pretty cool, folks, when you think about, you know, so we have, so say we look at, let's get an image here. Let's get an image. Yeah, I don't want to just talk about it. Let's look at an image. So let's look at, so extraocular eye muscles. And you're going to have to know this in the lab. And so here we go. This is a great image right here. Okay. I'll make it bigger. Okay. So this is the left eye, right? So as you're looking at, this is my left eye that I'm pointing to. Okay. Let me just get a sip here. All right. So if it's my left eye, okay, medial rectus, what's going to happen with this? So medial rectus would be then the inner, so close to the nose, right? So see nasal side, temporal side. So in the inner portion, right? So if the nose is over here, nose is here. So we have here medial rectus, lateral rectus, superior rectus, and inferior rectus. And then we have coming to the medial aspect, we have the inferior oblique and the superior oblique. Okay, and now just look at these muscles and imagine if they were to contract, and really they don't just contract on their own, they contract in conjunction with each other, so that this way you can, so if you look at me for a moment there and just see that I'm looking superiorward, I'm looking inferiorward, I'm looking to the left, I'm looking to the right, not moving my head, right? And then I'm looking, doing cross-eyed, that's a little wacky. But pretty cool though, right? That, you know, understand that all these extraocular eye muscles will work in conjunction to allow for smooth movement of the eyeball. And I control them so they're skeletal muscles, right? And so on the practical exam, you will need to know these muscles, okay? But yeah, they're small, they're tiny muscles, folks. You'll see in the lab that when you look at them, you're like, whoa, that's really tiny in comparison to say quadriceps and that group of 4 muscles that make up the anterior thigh. So let's come back here. So eye muscle contractions are rapid and brief. Gastrocnemius would be your calf muscle. When you think of your calf muscle, think gastroc is more the superficial, and then deeper would be soleus muscle. Okay, they contract more slowly and hold the contraction longer. And know that you know, when you're standing and such, I mean, those muscles really, they're different as far as their work and their action in comparison to these smaller muscles, which have brief action. Okay, so recall that I said to you that term, the action of a muscle. And oh, let me come back here for a moment and just show you this. That really, so imagine if this muscle were to contract, what's it going to do? It's going to pull the eye so that it's looking superiorward. Or when this muscle contracts and it pulls, right, it shortens, it's going to pull the eye inferiorward. And same here with medial rectus and lateral rectus, and working in conjunction with the oblique muscles, there's a lot of smooth movement that takes place with the eye movement and such. So very important to keep that in mind as far as the action of a muscle. And you can figure it out just by seeing a muscle and going, okay, well, if that contracts, what's going to happen? What part of the body of the anatomy is that going to move when it does its action? Okay. Now, the term tone, muscle tone, we all, as we're sitting here right now, and as I'm sitting here in front of you, my musculature is not, again, and I've kind of kidded around about this, but like, I'm not like, you know, like just like totally all my muscles into, you know, strong contraction. No, but there's a slight contraction of the musculature. There is, right? As a result, we say that this is tone, constant, slightly contract state of all muscles, and allowing for me to be erect and not just kind of slumped over, right? That's muscle tone. And our postural muscles, very important that they're able to do that. And we'll also talk in this semester regarding reflexes and such, and that's pretty cool stuff. Yeah, so we'll discuss that also as far as what takes place and how reflexes also help to protect the musculature, and very important aspect of our body's nervous system and muscular system working together. Now, recall that I said to you that when cross-bridge formation occurs, right? ATP is important in order to allow for separation of the myosin head from the myosin binding sites on actin. If that doesn't take place, if ATP is not present, you know, that's not a good thing, right? But rigor mortis, that's what takes place, because the body is not synthesizing any more ATP, and as a result, this body is dead, and the cells are not functioning like they properly should. They're not in a living human, and as a result, no ATP is produced. So the cross bridges do not separate. The myosin head and actin do not separate, and as a result, we have that rigor that takes place, that sustained contraction, that takes hours to really like a half a day or so, to closer to a full day, for the muscles to relax and to not be in sustained state of contraction. So ATP is the only source of energy and needs to be regenerated quickly. So mitochondria. So when you've seen images that I've shown you of the skeletal muscle, you can see that there are many mitochondria present, and they are important in order to produce the ATP. Oh, excuse me, one moment. Okay, so the production of ATP, very important, right? So you'll see here that aerobic respiration produces 32 ATP. So this is really, in the presence of oxygen, this is really the most important and longer lasting production of ATP that's very important. You'll see here that direct phosphorylation of ADP, right? So adding a phosphate to ADP, creating ATP. So this is ADP, 2 phosphates, ATP, 3 phosphates, right? And actually the breaking of the bonds between the adenosine and the phosphates that are attached, breaking that bond releases energy. And that's what's really why ATP is important as far as energy is concerned. The breaking of bonds releases energy. Anaerobic pathway, so we're having in the lack of presence of oxygen, glycolysis, lactic acid formation, so we're producing 2 ATP. So you see here, ATP is produced quickly, but not very much. But aerobic respiration produces many ATP. You see, anaerobic respiration yields only 5% as much ATP as aerobic, but again, produces ATP 2.5 times faster. So you need to, that's an important sentence that should stick with you, that you should know that, okay? You'll see here that as far as muscle fatigue is concerned, we get this, right? Physiologically, inability to contract despite continued stimulation, because you've depleted ATP and you're so tired and you've done so much work. So I think back to, in particular, so even when I did my working out with weights as a young man, and with my brother's only a couple years younger, and he and I would train for quite a few years, and we would work out hard, but we really, you know, like I can't say that we really like went totally to the point where I could hardly lift my arms. Never did that, but we worked out hard. But I can say when I was in the service and I was in boot camp, and they trained us so that pretty much you felt like you wanted to collapse. You know, you were worked so hard in the heat. I was in Cape May, New Jersey in the summer, and that physical training was just, that was brutal. And we were in hot rooms, doors closed, windows closed, and you're exercising, and it's just like a sauna. And you just feel like that's the time in my life where I can think of total physical muscular fatigue as a result of working and training and such. So that can happen, right? But you know, it all depends upon the load that you've placed upon the muscles. So you'll see here that as far as the different types of contraction of ATP production are concerned, okay? Again, phosphorylation and glycolysis. And again, these are only producing 1 and 2, right? ATPs, right? 1 and 2 ATPs. But then we see here, as far as aerobic respiration, right? We're seeing that 32 ATP are being produced. Takes longer for this to happen, right? Again, these take place much quicker, but produce only a little bit of ATP in comparison to what's going on as far as cellular aerobic respiration is producing the many ATPs. Now, this is important for you to see here as far as the different types of fibers that are present, okay, within skeletal muscle in order to really they're unique as far as what their function and role would be. Okay, so you need to keep this in mind as far as slow twitch, fast twitch oxidative, fast twitch glycolytic. So again, in the presence of oxygen, what's taking place? So as far as more long distance running, and then kind of medium and short. Okay, so sprinting and such. So you'll see here as far as slow twitch and fast twitch, and whether it's in the presence of oxygen or not, okay, we will allow for different types of fibers, and know that genetically you can be more predisposed to being more of a distance type of a person, having being able to swim longer, run longer, or more of a sprinter type of a person, just because of genetically the composition of your musculature. But know that we all have all of these types of slow and fast twitch, oxidative and glycolytic type fibers present within our musculature. But training, right? So training can really help to improve when we know this, folks, right? Because I can remember as a young guy, I didn't really like running, right? But I did, and I trained, and I was able to, by the time I got into the service, I was up to like 5 miles. But for me, that was a lot, because I really wasn't more of a distance guy. I was more of like, I like to sprint and such. But 5 miles was pretty good for me. That to me was like the marathon. After that, and I just could never get into really doing distance running. I couldn't. 5 miles was my limit. Like I think I got up to like 6 miles a few times, but that was it. That wasn't my thing. But again, I had to train for that. It took me a while to get to that point where I could do that many miles. Yeah, so training is in part takes a role in this, as well as genetics are concerned. And you'll see here that, so let's look here. So different muscle fiber types. So the slow oxidative, right? So we have here, as far as for endurance activities, maintaining posture, right? So that's slow oxidative. And then you'll see here fast oxidative, again in the presence of oxygen, medium intensity activities, so sprinting and walking. And as well, no, it's really, so the sprinting, I wouldn't put that so much as, because really it's kind of like below, like would be, when I think of sprinting, I think of really running hard. Okay, and so that's, I'm sorry, I shouldn't have put that there. So you could put that there as far as the fast glycolytic. I apologize. Please cancel that out and then just put that right here. But also, like another thing would be like hitting a baseball. That's a very fast type motion and such. And so intense, powerful movements and such, fast glycolytic. Okay. Now, look here for a moment. You're just seeing here also, as far as how skeletal muscle has a combination of slow and fast twitch muscles. So there's a combination of both of these types of muscle fibers present in skeletal muscle. And this is very important for you to see here. The hypertrophy, the increase in size of a muscle, right? It's not a matter of that there are more fibers, right? So look at this, see this muscle belly right here? And there are how many muscle fibers present, okay? Or actually, yeah, these muscle fibers present within a, oh, darn it. Oh, give me one moment. I do apologize. Fascicle, yeah, it's the fascicle. See the fascicles right here? So we're looking at the muscle fibers. So I'm looking at a fascicle on that other image. So that's a fascicle of many muscle fibers. Here's one muscle fiber composed of many myofibrils. Here's one muscle fiber composed of these, I'm sorry, I'm messing up now. I apologize. So the fascicle comprised of many muscle fibers. Here's one, these are the fascicle itself with many muscle fibers. Here's a muscle fiber composed of myofibrils, and a myofibril composed of the myofilaments. I'm sorry, guys, I apologize. So do you see what we're seeing here as far as these fascicles? This is a fascicle. This is a fascicle composed of these muscle fibers. Each of these muscle fibers are composed of myofibrils and then myofilaments. Understood? So that's what we're looking at here, is these different muscle fibers present in a fascicle. That's one fascicle with many muscle fibers. Okay, and so knowing that, again, fast twitch and slow twitch. But again, we're not having as muscle fibers, as the muscle gets larger, hypertrophies. It's not as a result of more muscle fibers. It's just that the size of the muscle fibers increases. And so what will happen is that there'll be more water present and more myofibrils and myofilaments. Okay. All right. Okay. I hope that wasn't confusing. I apologize. Sorry. It's just it's been a long day. I apologize for that. All right. So let me minimize this. Let me stop recording.